So how can remote live-cell imaging help? So first I want to show you this small video, or short video I should say, from Professor Robert Davey at Boston University. And this video is made by the WGBH News. And you see that the people here are working in a BSL-4 lab because of the positive pressure suits they're wearing. And they're actually using the Lux 2 system over there to monitor the cells that are infected with the virus. And this is one of the example videos that they showed. And as you can see here, by the appearance of the black spots, those are cells that are detaching because they are infected with the coronavirus. So remote live cell imaging can help these people because they can investigate their cells remotely via the cloud. And because of this, they don't need to enter the lab. So this makes it very time efficient because they don't need to spend 15 minutes to to put all the stuff on and also to exit the lab. Next to this, people can check their cells within just a few clicks online. And because of this, you don't need to do all the safety measures that you would normally take when you are working in the lab. This makes it a lot less effort than actually visiting the lab and checking your cells. Next to this, we can provide time-lapse imaging with remote live cell imaging. And because of time-lapse imaging, you don't have to get one sample for time point 1, one sample for time point 2, another one for time point 3. You can just use the same sample for each time point. And because of this, you need to have less samples, which means less disposables, but also less cells, of course, and also less protective equipment because you don't need to go to the lab for each time point. And this makes it also a lot more cost effective. Next to this, you get regular information about the cell status, especially when you're using the confluence algorithm, for example. And when you know exactly when your cells are ready for your experiment, you can also plan your experiment better and you have less waste of your materials and also your time. And this is especially important for people doing COVID-19 research at the moment because there's such a high pressure to find the right cure for this virus. People need to use every bit of time they have in an efficient way. So really gaining a lot of time using live cell imaging can help them out. Next to this, you also have less contact moments with the pathogen. And when you have less contact moments, you have a lower risk of contamination, so a lower risk of getting ill, which gives the people more peace of mind. So how can the Lux 2 and the Omni help in this situation? Well, first of all, we can investigate the cells remotely via the cloud in just a few clicks. So in this way, you save a lot of time and effort. Also, time-lapse imaging provides them information about the cell status, so they know when their cells are ready for an experiment. Next to this, they have less contact moments with the pathogen because our devices are sending all the images to the cloud, so they don't need to enter the lab. Next to this, one very important feature that not all live cell imaging devices have is that our devices can be installed without the help of an engineer. Normally, you would need to have an engineer of the live cell imaging company enter the lab and install the devices. And this is, of course, a very risky situation for both the engineer but also for people working in the lab because the engineer doesn't know how to work in these labs. So when you can install a device without the need of an engineer, this is a lot more efficient but also saves a lot of time for them and also a lot less risk of things going wrong. Next to this, our devices can be sterilized really easily, which is very important when you work in these BSL-3 and 4 labs. And the feedback we got from the COVID-19 researchers is that they can use live cell imaging in about 80% of all assays that they're doing. And some examples of these assays are, again, the cytopathic effect assay and the neutralization assay, but they can also use it to check when their cells are ready to be infected by the virus, or also what effect adding a virus has on the growth of the cells.